Hello, this is chapter 20. Whoops. And this is on phylogenies, which is like those sort of like family trees showing how different organisms and species are related. So we've seen these before, we've worked with them, but now we're going to formally learn how to use them. And this is how we organize life. And here is an example. So on the left, we have two different kinds here. This one is kind of an older version of how we used to do them, and this is called a rooted one, where we are looking at what might have been a common ancestor, splitting out into like 3 different groups here. So the blue group is bacteria, and the green group is archaea, and that's a separate domain. So we're looking at 3 domains, each color is a domain, and those are like the organisms that are in hot springs in like Yellowstone, and they're extremophiles, so they live in like really cold places, really salty places, really hot places. They live in extreme environments. Usually there are some that live in us. And then over here we have eukaryotes, and those are us. So here we are, the little star, and all these other organisms, pretty diverse. And one thing to note here is, as you can see, surprisingly, archaea are more similarly related to eukarya than prokarya. And prokaryotes. Both of these, or I should call these bacteria, sorry. Both of these groups, the blue and green, are technically prokaryotes. So we are seeing that the green ones, the archaea, which are single-celled organisms, no nucleus, they're more closely related to eukaryotes than they are prokaryotes. But both the green and blue do not have a nucleus, they're single-celled. And then the red can be single-celled or multi-celled. This unrooted tree is another way to look at it, where we're not really looking for a common ancestor, but we're just seeing how they're related in a little bit of a different way. So we'll use both of them. Obviously, if they're closer together on both, they're more closely related. So like we are more closely related to fungi than we are to plants, which makes sense because fungi does cell respiration like us, and plants do photosynthesis. So those are some different phylogenetic trees. And this is kind of the structure of it. So down here you have the root, and then when it branches off, that's a branching point. Down here, these are called basal taxon. And then when you have a whole bunch that, just wait, Erica. When you have a whole bunch, this is a polytomy. And then these are sister taxa when they kind of branch together. So those are some parts to a phylogenetic tree. So basically, here's some kind of definitions down here. If they're unbranched, it's a basal taxon. And if they stem from the same branch, it's a sister taxa. And if there's a whole bunch, it's a polytomy. So here's an example, kind of separating some of these vertebrates, those that are in the phylum Vertebrata. So the first thing is, do they have, some of these don't. Do they have a vertebral column? I'm not sure how I'm following this. The answer is no. This is strange. Anyway, they do all, these lanceolate do not. They're kind of on the cusp of vertebrates or not. But then it's showing if they have a hinged jaw, then you keep going. And so it kind of branches and categorizes them. Like, does it have hair? Yes or no? Oh, I see what it's doing. I'm sorry, you guys probably saw it before I did. So the lanceolates do not have a vertebral column, so they branch off there, but the rest of them do. And then do they have a hinged jaw? No, lamprey do not have a hinged jaw, but the rest of them do. Do they have legs? No, fish do not have legs, but the rest of them do. Do they have eggs with amnion? No, frogs don't have amniotic eggs, but the rest of them do. And then do they have hair? Lizards don't have hair, but rabbits do. So that's how you kind of use that. So here's another look at this, and I'm gonna let you guys watch this video because it won't let me upload a YouTube video if I have a YouTube video embedded. So you can kind of watch how we use this, but you can see like lungs here are going to the salamander, that sometimes they have a combination of lungs and gills. Amniotic membrane on this lizard, they start to get amniotic eggs and they can move away from water. Hair on the mammal. So that's how you would use those branching off points. And then this is how we classify them. And we've all seen this. We start with domain here at the bottom, and then it goes up to kingdom, phylum, class, order, family, genus, species, and you might have some kind of way to remember this, like King Philip can order 5 green sausages, something like that. And as we go up or down, we get more and more narrowed down to that species, and we can even have subspecies. Remember that genus and species are italicized. So you can see here Canis lupus. This is the subspecies familiaris. Canis is the genus. Lupus is the species, and then we have a subspecies, and those are all italicized. So then we know they're a scientific name. So here's another way to look at it. We start with domain Eukarya. We've got plants, insects, fish, rabbits, all kinds of things. We should have even little little algae. Animalia then narrows some of that down. Chordata is just those that have a backbone. Mammalia is just the mammals. Carnivora are the ones that are meat eaters. Canidae are the ones that are like more dog-like. And then Canis is in that genus. Lupus is in that genus, so wolves and dogs. And then the familiar subspecies tells us that it's a dog. So that's how we categorize them. Now for section 2, we're going to look at how are they related to each other evolutionarily. And so one thing that we look at for evidence for evolutionary relationships is homologous structures. And so what those are when you have different animals or different organisms that have like the same bones, but they just have a different shape. And so in this example, you have the humerus here on the bird, and then you have the humerus here on the bat. You have the radius and ulna. And here you also have a radius and ulna, and then you have the wrist bones and the phalanges. And so they have all the same bones, and they're hooked up the same way, they're just different shapes. That is evidence for relatedness. So that shows that they are related to each other. Analogous structures are the opposite. Analogous structures do not show that they're related to each other. So they have the same function, totally different structures, they're not related. So here we have this bee wing. That actually might be a fly because it has 2 wings. It might be Diptera. Anyway, this structure is totally different from the bat wing and bird wing. They do the same thing, but the structure is different, so we know that they're not related. That would be silly. So here's a nice graphic that shows that. In this case, all of these are the same bones, but they're all used for different purposes. So we have similar structure, different function. Here, these all have the same, like, let's see. Okay, these are all kind of crawling aquatic creatures, sort of swimming. I guess they're all swimming kind of fins. They have the same function, but different structures. So they're not very closely related. Okay, we can also use DNA and proteins to determine how closely things are related to each other. And so if we look at this phylogenetic tree, we can see how many differences there are in the polypeptide hemoglobin. So if there's a lot of differences, they're very different from each other. And if there's fewer differences, like between the human and macaque, then they're more similar. So we're looking at their genetic DNA and how close they are. And sometimes it's confusing when you build a phylogenetic tree off of like anatomy and their structures and their bones versus molecular. Sometimes they give us different answers. And I think in general, we would say that the molecular comparisons would be more accurate, but it's hard to be able to tell in that case what we're looking at if they're contradictory. So here's an example of that. Here we have genetics on the left with molecular comparisons, and then we have body plans on the right. And you can see what kind of differences there are between these. They are in not the same order. So like here we have Nematoda, and then on this side it's over here. Arthropoda, is here, and it's over here, and these are kind of mixed up differently. So when we look at the in fact, I would claim that the molecular comparisons are a little bit more organized, and then these look a little bit more mixed up when we're just looking at body plans. Okay, so let's move on to the next one. This is a phylogenetic tree of vertebrates and a vertebrata of the phylum. Lancelets do not have a backbone, but they're included because of other rules. And then this is the same that we saw before: lampreys, fish, lizards, rabbits, and humans. These all have amniotic eggs. This does not include fish. And then the rest of them are just amniotes. So you can see kind of where that branches off. And then These are called, when you have some organisms branching from one point on a phylogenetic tree, those are called clades. So like these is polytomy, they're branching from that same point and they're called clades. And then these are not clades because they're not branching from that same point. They're not branching together. Okay, section 3. This is going to kind of talk you through how they figured all this out. Charles Darwin was one who was kind of looking at how animals might be related. And a lot of people had a lot of ideas about this that they were trying to make sense of. And a lot of people were stumbling upon these ideas at around the same time independently, which is interesting. But he kind of conceptualized it as a tree, as branches on a tree. So he kind of looked at like the main stem or trunk, there we go, and that it can branch off like leaves, and that these organisms are related to each other more or less closely. And this kind of also aligns with Mendel's genetics because we're looking at how organisms are related to each other genetically. And the problem that makes this difficult is that sometimes genes can be transferred between unrelated species. That happens a lot with like bacteria. They can get up next to each other and transfer genes. Sometimes it's a gene to make them resistant to antibiotics, which is bad. But you can also have organisms exchange DNA that are totally unrelated, which is tricky, like COVID-19. COVID-19 doesn't integrate its RNA into the cell, but like HIV does. HIV integrates its DNA into the genome of the cell. So that's kind of tricky. Here's a picture of horizontal gene transfer. So we can see that two bacteria can get up next to each other and they can exchange DNA. And in this example here, this is an antibiotic resistance gene. So it's gonna give this bacteria resistance to antibiotics, which is problematic. So that makes the world a not good place. And then here's phages or viruses that do the same thing. And then here's just two cells exchanging DNA between each other. And then again, you can watch this video. I just can't play it on here. So those are some examples of how that works. Okay, thank you. This is how prokaryotes can exchange DNA. So transformation, transduction, which is bacteriophage, conjugation, and gene transfer agents like phage-like particles. Some of those I showed you in the pictures. And then eukaryotes can do it from food organisms. There are jumping genes that happen in like corn. Epiphytes, which are like parasitic plants, can input their DNA, and then viral infections. So I have heard that our gene to make placental protein, to make our placenta, comes from a virus long ago that made us placental animals, that type of mammal. So that's really interesting. Here's an aphid, and they get their pigment from a red carotenoid that they get from the plant. And so they're getting that from genes from the plant. So that's a horizontal gene transfer from the plant. Barbara McClintock is really famous. She was the first one to theorize about jumping genes, and she studied corn. So you know how corn like has different colors on it? There's different colors of corn. That's where she was working. And then another famous scientist, female scientist, I don't remember her name right now, but she came up with this endosymbiosis hypothesis. And this just says that this is where eukaryotes came from. So eukaryotes basically brought in these little bacteria, and they turned into mitochondria and chloroplasts. So that's the endosymbiosis theory. And then they theorized that this is where gram-negative bacteria came from, which is a specific type of bacteria. That archaea kind of enveloped gram-positive bacteria because the cell wall around gram-negative bacteria is a lot like archaea's cell wall. So it might be, they might be right. And then here's a video from Paul Anderson that you can watch that I have to skip. So here's a question that scientists are really pondering. What came first in terms of eukaryotes? So these are 3 models. Did the nucleus come first? So did a little cell with its genetic information, like a little prokaryote, did it move into a larger cell and then form a nucleus, and then things went from there, and that made like mitochondria later was integrated in the cell, another little bacteria? Or did the mitochondria come first and then later the nucleus was integrated in? Or was it first somehow a eukaryote and then the mitochondria left and became a prokaryote, like a bacteria? This one's probably the least likely, but these two are interesting. What came first, the nucleus or the mitochondria? So here's another view of that tree of life. But what they're showing here is that there's probably a lot of mixing and crossing between like bacteria, archaea, and eukarya. So there's probably a lot of borrowing of genes, like through horizontal gene transfer. And maybe it looked more like this. Maybe this was more the accurate model, where there were all these primitive cells and then they all branched off kind of equally to make different types of cells. Okay, there's another video there if you'd like to watch that. This will switch to the next slide. Oh, that's our last slide. So hopefully you watched those videos and hopefully that was helpful for you.